Here comes the next topic, Mendel's dihybrid cross. So till here we have seen that he has been taken the example of monohybrid cross. That means he was taken only the single character. That means a single character in the case of height, tall and dwarf. He was crossing with a single character, right? But yeah, for the next experiment, he is going to take up the different example and he is going to cross up with the two characters. That means as the name indicates, dihybrid, di means two. That means two types of character is going to combine to get the dihybrid cross, and we'll see how the Mendel has been proved the dihybrid cross with the help of pea plants. What are all the characters he has been taken to prove this? So the Mendel dihybrid cross is also known as inheritance of two gene. Mendel dihybrid cross is also known as inheritance of two gene. So the statement of the dihybrid cross states that the cross between two parents of contrasting character is known as dihybrid cross. The cross between two parents with two pair of contrasting characters is known as dihybrid cross. Remember well, in this he is going to take two parents with two pair of contrasting character. In the monohybrid cross we were seeing only one pair of contrasting character, that means tall and dwarf, right? But in case of dihybrid cross, is going to take two pairs of contrasting characters. That is known as dihybrid cross. Or it can also be defined as if two allelic characters are used in a cross, then it is known as dihybrid cross. So in case of monohybrid cross, we were seeing that he is going to cross only capital T and small t. That is a single character which is being crossed over there. But to prove this Mendel dihybrid cross, he has been taken the two parental generations and two contrasting characters. So the example taken by Mendel to prove this Mendel dihybrid cross is, he has been taken the characters like shape of the seed and color of the cotyledon. The character is being taken over here is characters shape of the seed and color of the cotyledon. Yes, shape of the seed and color of the cotyledon. These are the two contrasting characters taken by Mendel, are selected by Mendel to prove the dihybrid cross. So the shape of the seed and the color of the cotyledon. So what is the shape of the seed and what is the color of the seed and what are the characters is being selected for these two characters? Let us see now. So the shape of the seed is going to take round and wrinkled. He is going to take round and wrinkled, and he is going to say that the shape of the seed, both round is going to play the dominant role, that is capital R and capital R. Wrinkled is going to play the recessive character such that it is represented as small r and small r. Remember well, the shape, the first character of the dihybrid cross is going to take as the shape of the seeds, and the shape of the seeds is of two. One is round and wrinkled. The round will be the dominant character where it is being represented with small capital R and capital R. Wrinkled shaped seeds, these are the recessive characters, so such that is going to take smaller and smaller. This is one character. In the dihybrid cross, is going to cross with the two characters, right? So the shape of the seed is a one character, and the other character will be color of the cotyledon. Cotyledon is nothing but seed, right? So the first character is shape of the seed and the second character is color of the cotyledon or the seed. So it's going to take with a yellow and green colored. So it's going to take up with the yellow seeds and the green seeds. So that is being taken two characters. One is shape of the seeds and the other one is color of the seeds. That means two characters of the two different parents. That means round is different parent, wrinkled is different parent in the case of shape of the seeds. In case of the color of these seeds, here we can see the two parents. One is yellow, other one is green, right? Such that he has been mentioned with the genotypes too. The dominant character with the capital letters and the recessive characters with the small letter. Please don't get confused. He has been taken two characters with two different parents. That means one is shape of the seed, other one is color of the cotyledon. Shape of the seed is round and wrinkled, and color of the cotyledon is yellow and green. So as we know now, for the dihybrid cross, the Mendel has been selected two characters. He is going to select two homozygous pea plants. Two homozygous pea plants, one is male, other one is female. So it will be the homozygous plants, such that the homozygous plant is going to select is two. There should be two characters. Remember well before as I told, here is going to select homozygous round yellow seeds. Round is represented as capital R, yellow is represented as capital R, capital Y and capital Y, such that this round yellow character is going to play as the dominant character. This round yellow character is going to play as the dominant character. So this will be the homozygous male plant selected for the dihybrid cross. So the next will be wrinkled green seeds. That means wrinkled represents small r and small r. Green represents small y, such that it is, as the symbol indicates, due to it is written in the small letters, it is all known that it is a recessive character. So here the parental generation is ready now. He is going to select two characters. Round is other character, yellow is other character, wrinkled is other character, green is other character. For the dominant character, round yellow is selected. For the recessive character, wrinkled green is selected, right? So once after taking the example of these characters, dihybrid characters, di means two characters with the two different parents, let us see how the Mendel is being proved the dihybrid cross, or how he is going to get experiment results by the help of these two characters. So next will be Mendel dihybrid cross. Mendel dihybrid cross. As I told you before, the Mendel dihybrid cross, it is going to represent with the two parents with the two different characters, right? So the two different character is Mendel selected was round yellow plants, other one was wrinkled green plants, as green seeds, right? So the parental generation is going to select male homozygous round yellow plant, female homozygous wrinkled green plants. So the homozygous round yellow plants, it is represented by capital R, the capital Y. Here the recessive wrinkled green character, it is being represented with small r, small y and small y. So now after the parental generation taken here, we have been got the genotype. Mendel has been got the genotype where it's been represented. Then once after getting the genotype, it should undergo process called meiosis. It should undergo the process called meiosis, such that after the meiosis process only the gametes can be formed. So after the meiosis process, here the gametes have been formed over. So capital R, capital Y. These two are different characters. So from each character, a single allele or the single gamete should be passed on to the next generation, such that from these two characters, one single gamete is been selected. From this character, one single gamete is selected. Here comes capital R and capital Y. The same way in the recessive character, this is different character, this is different character, such that small r and small y is being selected, such that they are going to form the gametes over here. Carefully observe over here how the two characters are being mixed, and the two characters are going to play their role by self. And for single character, there will be always. So here he has been taken the genotype, as we have known that here he has been taken the two characters and the gametes have been formed over from that. So after the formation of the gametes, it should undergo cross fertilization or the cross pollination. So that the gametes after the formation, the cross pollination, here you have been seen the two characters, right? So each one is having its own character, such that capital R is carried over here and the next capital Y is carried. Here comes the small r, it's carried over here, small y. Such that capital R represents it represents the dominant character, it will be round, and small r represents wrinkled character, represents recessive character. Capital Y represents dominant character, yellow color of the seeds. Then small y represents the green color recessive character of the seeds, such that The result of the formation of the gametes of these two by the cross fertilization process, we have seen the formation of the F1 generation seeds. We have seen Mendel has been proved the formation of seeds of the F1 generation. F1 generation, the result is capital R, capital Y, small y. This R and R represents the shape of the seeds. Shape of the seed will be, because of the presence of the dominant character capital R, it is going to say, phenotypically the plant or the seed will be always round. Always will be round, remember. Then the presence of this color of the seed will be, due to presence of the dominating character capital Y, it will be represented as yellow. Such that the combination of these characters, the first F1 generation of the dihybrid cross will be round yellow seeds plants. Round yellow seeded plants. So this is a result of the F1 generation proved by the Mendel by taking the dihybrid cross. So once after the formation of the F1 generation, we should undergo it. It has been undergone the F2 generation by self-crossing the F1 generation plants itself. So by self-crossing the F1 generation, is going to take parental. It is going to take represented as parental generation 2. So in the parental generation 2 is going to take up with the F1 generation. That means these are all not the homozygous plants, right? These are all the heterozygous plants, such that male heterozygous round yellow plant is crossed with a female heterozygous round yellow plant. So this will be the parental generation. We should know the genotype for these plants, right? So this will be the genotype, that is capital R, capital Y, small y. The same way for both the male and female, same F1 generation plants has been taken over here to cross fertilize. So once after the genotype has been formed over here, once after the formation of the genotype here, now it should undergo the formation of gametes, right? So here comes the complex thing. Once after taking the genotype of the dihybrid cross, it should undergo the gametes formation. Then how the gametes will be formed over here? Are you observing? There are the two different characters. That means the color of the seed and the shape of the seed and the color of the seed. So in the shape of the seed itself, there are two characters. One is round, other one is wrinkled. So all the 4 different gametes are going to carry the different characters. Then how can the gametes can be formed? Let us see now. So this capital R hold on with the dominant character. So this can be crossed with this is single character. ಇದು ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಆಗಿರುತ್ತೆ. ಅಂದರೆ ಇದು ಇಲ್ಲಿ ರೌಂಡ್ ಅಂದರೆ ಕ್ಯಾಪಿಟಲ್ ಆರ್ ಸ್ಮಾಲ್ ಆರ್ ಇರೋದ್ರಿಂದ ಇದು ಏನಾಗಿರುತ್ತೆ, ಇದು ಒಂದು ಕ್ಯಾರೆಕ್ಟರ್ ಬಂದು ಶೇಪ್ ಆಫ್ ದಿ ಸೀಡ್ ಅಲ್ವಾ, ಇದು ವೈ ಬಂದು ಕಲರ್ ಆಫ್ ದಿ ಸೀಡ್. ಆದ ಕಾರಣಕ್ಕೆ ನಾವು ಡೈಹೈಬ್ರಿಡ್ ಕ್ರಾಸ್ ಅಲ್ಲಿ ಎರಡನ್ನೂ ತಗೋಬೇಕು, ಕಲರು ತಗೋಬೇಕು, ಶೇಪು ತಗೋಬೇಕು. ಆಗ ಮಾತ್ರ ಎರಡು ಕ್ಯಾರೆಕ್ಟರ್ ಮಿಂಗಲ್ ಆಗಕ್ಕೆ ಸಾಧ್ಯ. ಸೊ ಫಾರ್ ದಿ ಗ್ಯಾಮೆಟ್ ಫಾರ್ಮೇಶನ್ ಆಫ್ ದಿ F2 ಜನರೇಷನ್, ಹೌ ದಿ ಗ್ಯಾಮೆಟ್ಸ್ ವಿಲ್ ಬಿ ಫಾರ್ಮ್. ಸೊ 2 ಕ್ಯಾರೆಕ್ಟರ್ಸ್ ನ ತಗೋಬೇಕು ಅಂತಂದ್ರೆ ಇಲ್ಲಿಗೆ ಒಂದು ನಾವು ಆರ್ ನ ತಗೋಬೇಕಾಗತ್ತೆ. Here the gametes are being formed over here. Are you observing that we have taken the 2 different characters? One is round, other one is this will be the shape of the seed and this will be the color of the seed. So these are the 2 different characters, that will be round and this will be wrinkled, such that now I am going to show how the gametes will be formed due to presence of these two different characters. First take the dominant character capital R. This will be one character. Then we should select the other character for the formation of the gametes, right? So this is one character, this 2 will be the other character. From this I am going to select the capital Y first. So these 2 we are going to form the one gamete. Next, this capital R is going to combine with the small y. Capital R and small y. Then here, have you seen this capital R and capital Y? Then this can be crossed with this character capital R here and the small y. Next, this is remained over here. This small r is crossed with capital Y and small r is crossed with small y. So this is how the gametes have been formed over here. So the same procedure is being followed again for the next plant. Capital R, capital Y, capital R small y, small r capital Y, small r small y. So this is how you're going to get the 4 different gametes for the F2 generation. So this is how the gametes are being formed from the F2 generation here. As capital R, capital Y, small y, capital R, small y, small r, small y, and the same thing is repeated for the second plant. That means a male heterozygous plant and the female heterozygous plant. We have been got the 4 different. So after the formation of these gametes over here, it should undergo the cross fertilization between these gametes, right? So the F2 generation cross fertilization, due to presence of so many gametes, here we can take the help of Punnett square, such that the male gametes are being arranged over here, then the female plant gametes are being arranged over here, such that the cross fertilization take place. Here the capital R, capital Y, capital R being written, that is the shape of this seed, and the capital Y represents the color of the seed. By the cross fertilization of these 2 gametes, we are going to get the seed which is round yellow as the dominant character, and as per the recessive character we can nominate the shape of the seed, that means round yellow. In such a way you can cross verify or cross pollinate all these gametes, and we are going to get this cross checkerboard. Here totally we are going to get the 16 crossing overs from the F2 for the F2 generation from the F1 hybrid plants. F2 gametes are 8. At the result of the F2 generation crossing over, we are going to get the 16 plants result. That means Mendel has been got the 16 plants result. Once after cross verifying, that means capital R, capital Y, then such a way he is going to cross all the plants. From these gametes, all these 4 gametes, from this gamete to 4 gametes is being crossed, and you are going to get the same result. Then from these gamete, all these 4 again, from this gamete, all these 4 again, and from this gamete, all the 4 again. So after the cross verify or cross pollination, we are going to get all these results. Then after getting the results, so here comes, as the gametes present over here, presence of capital R, Y represents round yellow. Then here comes capital R, small r, capital Y. As we know, capital Y is dominant character, of course comes the color yellow. But presence of capital R and small r, here the capital R is a dominant character, such that it's going to play the phenotypic form will be the represented by the dominating character. So the character of this plant will be the round yellow. As same it goes on, the presence of the dominating character, it might be ಇಲ್ಲಿ ಎರಡು ಅಂದ್ರೆ ಡಾಮಿನೇಟಿಂಗ್ ಕ್ಯಾರೆಕ್ಟರ್ ಮತ್ತು ರೆಸೆಸಿವ್ ಕ್ಯಾರೆಕ್ಟರ್ ಇದ್ದಾಗ ಡಾಮಿನೇಟಿಂಗ್ ಕ್ಯಾರೆಕ್ಟರ್ ಯಾವುದಿರುತ್ತೋ ಆ ಗಿಡ ಅದೇ ಆಗಿ ನಮಗೆ ಗೋಚರಿಸುತ್ತೆ. ಅಂದರೆ ನೋಡೋದಕ್ಕೆ ಹಾಗೆ ಕಾಣುತ್ತೆ, ಜೀನ್ಸ್ ಬೇರೆ ಇರ್ಬೋದು, ಆದರೆ ಕಣ್ಣಿಂದ ನೋಡೋದಕ್ಕೆ ಅದು ರೌಂಡ್ ಆಗಿನೇ ಕಾಣುತ್ತೆ. ಸೊ it is round yellow. So the gametes may be different, but the phenotypic will be different and genotypes are different, right? So here you can observe many plants are of having different genes but representing the same phenotype. As we go on, in this side also we are going to get the same phenotype, but when we are going to cross over here, we are going to get the different phenotypes, right? Small r going to give small r, capital Y going to give this, and the small r, it is a recessive character, such that it is going to represent the wrinkled character of the plants, such that it will be wrinkled yellow. So in this way we are going to get the checkerboard result. Then once after getting this checkerboard result, we want to write the phenotypic and genotypic ratio. It's quite difficult to opt the phenotypic characters and the genotypic characters. So to make easier, I have written in the green color, right? Whatever the characters have written in the green color here, capital R, capital Y represents, that means round yellow. It is going to represent a round yellow, right? Then here represents the wrinkled yellow, that is the character I have written in the red color. This is, here again comes the round yellow which is being written in the green. Then in the red what I have written, wrinkled yellow. Then after that, whatever the green color I have written, it is all of the round yellow. Red color represents wrinkled yellow. Then after coming here, capital R, capital Y, small y, it is capital R represents the dominating character of the round character, such that round, and this, it is a recessive character, such that it is going to represent the green color of the recessive plant. So it will be capital R, that means capital R representing round color and Y representing the color of the green. So it will be round green color seeded plants. So these three will be the round green plants. At the last one, here all are recessive characters. All the gametes or all the genotypes is completely recessive kind, and the recessive plant what we have got in the F1 generation was wrinkled green plant what we have taken in the parental generation, that means parental generation 1, such that we can that means Mendel have been observed that even though the characters which is having the recessive character are being suppressed in the F1 generation, they are going to be represented in the F2 generation itself also. That means wherever the alleles are present, those cannot be said as, even though it is being suppressed, we cannot say that the genes are being neglected or the genes are being discarded from the nature. It will be represented in one or the other generation. That means dominating character will always be represented, but the recessive character might be suppressed in one generation and it will be getting in the next generation too. So this is how the dihybrid cross is being crossed over here. After this, we should get the phenotypic ratio and the genotypic ratio, right? So here we can observe that round yellow seeds 1, 2, 3, 4, 5, 6, 7, 8, 9 round yellow phenotype plants are we can observe. So then the wrinkled yellow 1, 2, 3 wrinkled plants, and round green plants, round 1, 2, 3 round wrinkled plants, and other one is wrinkled green plant. Such that after this we are finalizing the phenotypic ratio of these dihybrid cross. We are going to get the ratio as 9:3:3:1. That means 9 are round yellow plants, then 3 other are round green plants, other 3 are round wrinkled yellow plants, then other last one is wrinkled green. Last one plant is wrinkled green. Okay, this might be the phenotypic ratio. Then what about the genotypic ratio? Different genes are being observed, or different genes are being arranged over here. So the genotypic ratio of these dihybrid cross will be 1:2, 2:2, 2:4:1:2, 2:1:2, 2:1. That means the different genes what is being seen here, all the different genes are being categorized and the ratio is being calculated. So how this ratio can be cross verified? That means if you count all these characters, the total number of the characters should be 16, and the total result of the this ratio should be, at the end it should be 16. That means 9 plus 3 plus 3, 12 plus 15 plus 1, 16 again, right? 1 plus 2 plus 2, 4 plus 4 plus 1 plus 2 plus 1 plus 2, total overall it gives the 16. That's what we have got, the 16 different new plants in the dihybrid cross. So this is how Mendel has been proved the dihybrid cross. So this is a very important question carried over in your exams. It might be asked for 5 marks. That means to prove the dihybrid cross with the help of Mendelian experiments. He has been taken the round yellow and the round wrinkled green plants as a characters. That means 2 characters of 2 different parental generation, and he has been proved with the F1 generation and the same F1 generation plants is being cross pollinated to get the F2 generation over here, and the phenotypic ratio and the genotypic ratio will be the same. So after this, getting the dihybrid cross, we must undergo, or we must learn about the test cross, what we have been done in the monohybrid cross. In the monohybrid cross, we have done with the test cross. That means to check out or to cross verify how the results are being got for the monohybrid cross. In the same way, here the Mendel has been experimented, or Mendel has been calculated the test